Hello and welcome to Chapter 11 review video for the AP Bio test. I am here with Kitten Penelope, who may or may not speak up as we go through this. Alright, so you must know the terms associated with genetic problems. P, F1, F2, dominant, recessive, homozygous, heterozygous, phenotype, and genotype. You guys are actually really good at this already, so I'm just going to briefly go through them one more time. We talk about the P generation, that is going to be your parental. F1 is going to be filial 1, or first generation after the parents. And then F2 is going to be 2 generations below the parental generation. Of course, the Fs can go on to F3, F4, F5 filial generations, but we really only deal with F1 and F2, at least in this class. But you can think of it as, if you want to think of it as the P generation are your grandparents, the F1 generation are your parents, and then you're the F2s. That sometimes helps kids keep straight who's who in generation terms. So P is parental, F is filial, and then the filials have numbers 1, 2, 3, and so on. Dominant and recessive, you guys are good. That's with regard to alleles or genotypes. We can have a dominant allele represented as a capital letter, recessive allele is a lowercase letter, and then the phenotype, being the dominant phenotype, would be an expression in traditional Mendelian genetics of either homozygous dominant alleles or the heterozygotes, where they would have one dominant and one recessive allele, but that would show the dominant condition. Homozygous, homo means same and hetero means different. So homozygous means that you have 2 copies of the same allele, and heterozygous means that you have 2 different copies. Phenotype is what you phenotypically look like, and genotype is what your genes are. I do want you to remember that genotype or phenotype rather, can be more general than usually we recognize. Oftentimes we think phenotype, we think something that we can see physically, but phenotype can also be things like behavior or molecular phenotypes that are an expression of the genes, but not necessarily something we can physically see. So kind of be a little bit more general with that term phenotype than just physical appearance. Okay, so we're going to go ahead and put some of this into context with this figure. And in this case, you can see that we have our P generation, our F1, and our F2 generations. And up here we have 2 true breeding parents, and true breeding just means homozygous. And so they appear purple and white, and then the genetic makeup, so then that genotype is going to be capital P versus lowercase p. Because they're true breeding, meaning they are homozygous, they only make one kind of gamete, and so we only have capital P for the purple flowers and lowercase p for the white flowers. Because of that, when these 2 parents contribute to the next generation, we call that one F1, everybody is going to have this purple appearance, this dominant phenotype appearance, even though all of them are actually going to be heterozygotes. Now that heterozygote flower, when it goes through the process of meiosis, half of its gametes, with regard to this particular allele, will have the capital P and half with the lowercase p. When we deal with this F2 generation, what we're assuming is that this same generation is being used for both, here we go, the sperm and eggs for this F2 generation. And so because of that, this is how we get that very traditional 3-to-1 ratio when we talk phenotypes. Notice that when it comes to genotypes, though, the ratio is actually 1 to 2 to 1. Okay, 1 to 2 to 1. And the 1 to 2 to 1 is just saying we have 1 homozygous dominant, 2 heterozygote, and then 1 homozygous recessive. Now, the 3 to 1 again is going to be that phenotypic ratio, but the genotypic ratio is going to be this very traditional 1 to 2 to 1 ratio. So that's just to put some context on those vocab words from the last slide. All right, and then this is also just showing how those phenotypic and genotypic ratios are derived. Again, showing that when we deal with the phenotype, these 2 right here are the ones that switch because, or the ones that are a part of this 3 ratio on the phenotype, but then would be separated out on the genotypic side. And so, we can call all these guys carriers if we want, when we're talking about that hidden allele, that hidden recessive allele. But ultimately what it comes down to is that if that dominant allele is there, it's going to be expressed, and that's why we have the different ratios between the phenotype and the genotype. Okay, so now what this is showing you is how do we figure out, if we've got an organism with a dominant phenotype, how do we figure out what its genotype is? Because it could be either homozygous dominant or it could be heterozygous, and that could matter. And so what we do is we do what's called a test cross. And what the test cross does is takes this unknown genotype organism and cross it with a recessive phenotype. Well, if it's recessive phenotype, we know it has to have this genotype, lowercase p, lowercase p. So then what we do is we take a look at the progeny. And by looking at the progeny, we're able to determine what the original parent was, and consequently, what the possibilities are for the children, or the progeny. So if the purple flower was capital P, all we would get would be purple flowers. Okay, because all that the white flower can give is that recessive phenotype, or the recessive allele rather. In contrast to that, if we were to get some white flowers out of it, we would know that we would have to have that phenotype having come, or that genotype having come from the parent plant, because otherwise we couldn't have white flowers who would need to get one little p allele from each parent. So by looking at these offspring, we're able to draw some conclusions about the genotypes of the parents. And that's called a test cross. Okay, you must know how to derive the proper gametes when working a genetics problem. So we're going to go back and look at a couple of those figures again, just very briefly. So here, the reason that we only had one kind of gamete on each side is because we were homozygous on both sides. Yet here, we had two different gametes being formed because we were heterozygous. And so half of our gametes would be big P and half of our gametes would be little p. That's what we then put into the Punnett square. This does lead me to a pretty easy or a handy trick when it comes to doing Punnett squares. We're very used to seeing, the 2x2 Punnett squares, or when we're doing dihybrid crosses, the 4x4s. But in reality, if you're only making one gamete, you don't have to do all those squares. So up here, if we wanted to do a Punnett square for this, we know we only have one kind of gamete. We know that every time we go through meiosis, we're going to get capital P and then we have little p's. We don't have to do this 4 times to know, oh, everybody's the same, 100% are going to be this. Well, instead we can say, and we would not even do this because we do it in our head, we would have big P as one gamete, little p as the other gamete, and just have that one box that would look like this. What's more realistic is to say, okay, if I have a homozygous dominant parent, big P, but I only have a heterozygous other parent, so let's use this one here, get rid of that. Well, we don't have to write this out twice because it's just going to be the same in the top row and the bottom row. And so it's just a time saver because it will all work out the same. Here I have 2 to 2, whereas if I hadn't done this bottom row at all, I would have 1 to 1, which is the same thing. So just kind of keep that in mind when you're doing the Punnett squares. Avoid the repetition. Be smart about it. You're on a time crunch. And so if you're only making one kind of gamete, when you're making your Punnett square, only give it one row or one column just to save time. Same thing here. To follow on what I was saying earlier, this Punnett square is kind of a silly one. This one right here is kind of a silly one because it's 2 of the same genotype, when in reality all we have to do is say, okay, all of my egg alleles are going to be big P, all of my sperm alleles are little p, everybody's going to look like this, not even negotiable. Same thing over here. If this is the unknown, we don't really need to do this side of the Punnett square over here, because we're going to end up with a 1-to-1 ratio, which exactly is what the outcome would be. So, back to figuring out how the gametes are being formed, remember, it's just, if that's your phenotype, half of your gametes are going to have this one, and half your gametes are going to have that one. And so that's how your gametes are formed, but use that information to help you be smart about your Punnett squares. All right, you need to know the difference between an allele and a gene. I'm not even going to show you guys this. you guys are pretty good about using them in the correct context. A gene is actually a region on the DNA molecule. An allele is a variation of the gene. And so if you would never say the allele for hair, you would say the allele for hair color. And it would be brown or blonde or something like that. But a gene is actually referencing a specific spot on a molecule, and the allele is talking about the variations of what's happening at that spot. You need to know how to read a pedigree. All right, so these are pedigrees. The reality with reading these things is that it really just takes practice in trying to figure out tricks for reading pedigrees. For me, what I look for are two parents, so like here, that are the same, that have offspring that are somehow different from them. And so here again, two parents that are the same that have offspring that's different for them. And at that point, then I can say, okay, well, if both of the parents have it and half of their kids have it, like we have there, well, what does that tell me about the affliction? And so in this case, if they're both have it and one kid has it, then that leads me to believe that that's going to be a dominant thing, because if it were a recessive characteristic and both parents have it, then all of their kids would have to have it. And in contrast, when we look over here, okay, neither parent has it, and then one of the kids have it, then that's going to tell me that that's recessive. And so that's usually what I look for in a pedigree, are two identical parents and then kids that do something unlike their parent, and then kind of working backwards from there. There's plenty of practice problems in your book as for all of these genetics things, and the answers are in the back of the book. So it's a really good idea to just familiarize yourself with the practice problems that are in chapter 11, just to make sure that you can do these. It's kind of like multiplication. The more you do it, the better you get at it. Here's your key. You want to take a look at this. Boxes are males, circles are females, colored in mean that they're affected. It's a good idea for you to try to remember that. That said, College Board, on all the questions I've ever seen, College Board will give you a key. So if you have a brain fart and you can't remember, look to see if they just tell you in the directions what's what. They're usually not trying to test that kind of, minutia, and so they'll tell you. But it is a good idea for you to try to remember the boxes are the males, the circles are the females, and so forth. All right, you need to be able to use data to determine Mendelian patterns of inheritance. Here's what I'm going to tell you. We're going to look at figure 11.8, but there are some fantastic problem solving tips on page 225 of your textbook. So it's well worth your time to just spend, 5 minutes refreshing your memory as far as those are concerned. So here's a fun diagram. Here's a dihybrid cross over here. I have yet to see College Board require you to do this. It is extremely time intensive, but I want to make sure that you've seen it. If you find yourself doing a 4x4 grid and counting genotypes and phenotypes, take a step back and see if you can be smarter about it, either by reducing your columns or your rows because the gametes are the same, or because it's a known ratio. So you want to keep that in mind as well. I chose this image to put on the slide because it has this 9:3 to 3:1 ratio. Kids get this wrong a lot. So for now, ignore this whole dependent assortment business and this independent assortment business. It was just in that diagram. But what I want you guys to realize is that this is looking at the F2 generation. Okay. And so what we don't have is a situation where you would ever get 9:3 to 3:1 and a F1 generation. So for example, if we have our true breeding parents and our F1 generation is all heterozygous, and in this case this is a dihybrid cross, which means we're dealing with two different traits, then when we cross those heterozygotes into making our F2 generation, that's when we get this 9:3 to 3:1 ratio. And that 9:3 to 3:1 ratio is a phenotypic ratio. And so what that means is that we have 9 of, let's see, so we have, they're telling us right here, 9 of these, so we have the yellow rounds, 3 and 3 each of the heterozygous, or the one dominant and one recessive trait, and then we have one that is the recessive trait for both of those, or the recessive alleles being expressed for both of those traits. So This one is expressing dominant color, and dominant wrinkleness. This one is showing dominant wrinkleness, which is none, and then the recessive color, okay, dominant color, recessive wrinkleness, and then recessive. It's phenotype, it's not genotype. I don't even know what the phenotypic ratio is for genotype in this. I mean, we could go through and count. It's probably like 1 to 2 to 2 to 4 to 2 to 2 to 1 or something like that. But the 9 to 3 to 3 to 1 is phenotype. Over here, we're looking at genotype, and the 3 to 1 phenotype. And this is just going back to what I said at the beginning about ignoring this bit about dependent versus independent assortment. We'll deal with that a little bit later on. Okay, but that's where those ratios are coming from. You want to familiarize yourself or remember what the ratios are and when they come into play. Okay. You need to know how to identify things that don't follow Mendelian rules of dominance. The diagram I'm using for this one is a summary diagram that's in the chapter text, or at the end of the chapter text, so page 224. And so this is right here. This is what we call our traditional Mendelian genetics, which is just saying everybody gets 2 alleles for one trait. One is dominant, or the trait could have a dominant or a recessive allele, and we have very simple, if you have one dominant and you look like the heterozygote looks like the homozygous dominant. There we go. So this is typical. Everything below this is going to be non-Mendelian features. And so here we have our incomplete dominance. And incomplete dominance, that's the one that kind of gave some credence to that blending hypothesis, and the blending hypothesis just being that we have a heterozygote that looks like a combination of both the dominant features as well as, or I shouldn't even call them dominant. The heterozygote has both a phenotype that is intermediate between both homozygotes, and so that's what we're seeing here with the pink. Codominance is when both are being fully expressed. And so we talked about blood types with regard to that, where if you were A blood type, you had the pokies. If you had the B blood type, you had the rounds, in this cartoon illustration anyway. And it's not the same thing as this. If it was the same thing as this, then on your blood cell, you would have something that had like a bottom base, and then a poke, and then a bottom. We aren't mixing the two phenotypes together. Here we're combining the two phenotypes. Here we have both of them independently being expressed. Blood type is also a good example for multiple alleles, and that's just saying that, everybody has 2 alleles and 2 alleles only, but the trick is that in the population you could have way more than 2 alleles. And so blood types is really an easy one to know, because we know is through conversation that you can have the A blood type, the B blood type, or the O blood type, and you get 2 alleles for those regardless. And then we had pleiotropy, and pleiotropy is when one gene can affect many phenotypic characteristics. And so that's, here they're giving you the example of sickle cell disease. I think we talked about cystic fibrosis in class, where it's like you can just get one gene and it really changes a lot about you phenotypically. When we're dealing with more than one gene, so 2 or more genes, further extensions of Mendelian genetics include epistasis, and then polygenic inheritance. And so epistasis is just saying sometimes you're going to have one gene that is going to control the expression of another gene. And we talked about the labs in that case and where the yellows come from with regard to labs. And just to say, if you are a yellow lab, it's because you lack the ability to express color at all. And there's no such thing as yellow color in labs. What it is a lack of ability to express color. The only 2 colors that can be inherited by a lab are the brown and the black. So all labs have a gene for either brown or black, none of them for yellow. However, we also have this question of whether or not they can express the gene at all. And so if we have little e, okay, that's the recessive condition for being able to express. And so if you're getting 2 copies of that little e, that's where those yellow labs are coming from. And so if anybody who's ever bred labs can tell you, if you have 2 yellow labs, they can only have yellow labs, but if you have 2 black, or you have a brown and a black, then, or 2 browns, you could get any colors in the world. And that's exactly what's happening, and that's what this diagram is showing you. Polygenic inheritance is just saying it's possible that one phenotype can be influenced by many genes. And skin color is a good one because it exists on gradations. And so the easy way to recognize this is when there are some kind of gradations of phenotype. So skin color, okay, we could line ourselves up as a class on a continuum from left to right in the classroom, from lightest skin to darkest skin, or vice versa. And the reality is the reason we exist on that gradation is because there are many genes that are influencing the color of our skin. Same thing is true for height. There's many genes that influence our height, and that's why we exist on a gradation or on a spectrum for height. So that's just your quick review of Mendel and Mendelian genetics. I hope you found it useful. Good luck with your quiz.